Good morning, students, parents, graduates, and distinguished guests. My name is Anita Chetty. I'm the Upper School Science Department Chair, and on behalf of Harker's K-12 Science Departments and the Women in STEM Club, I'm thrilled to welcome you in person to the 16th Annual Harker Research Symposium. Back in March of 2019, the 15th Annual Symposium was poised for another exciting event. But I'll never forget the Upper School Division Head, Mr. Butch Keller. I'll never forget his voice coming over the PA on a Thursday afternoon announcing the school would close for in-person instruction until further notice. The following Monday, we pivoted to remote learning, reaching out to our students in their bedrooms, their kitchens, and their backyards. It seems like there were an awful lot of dogs, cats, and stuffed animals that joined our classes. And now that I think about it, they actually went to Harker tuition-free for about 2 years. When in-person instruction ended for our students, so did the planned summer research internships at colleges and universities. But surprisingly, we soon realized that COVID-19 might have stalled in-person opportunities, but it did nothing to curb our students' infectious curiosity and feverish enthusiasm for exploration. Somehow our students managed to conduct meaningful research at home, and last year, I didn't think we would do it, but I was actually compelled to host a very successful virtual symposium. But today, students, we are all gathered here in person after 2 years to celebrate you and your discoveries. And we want to applaud all of your efforts. I'm very happy to say that for the first time ever, we have over 60 poster sessions, a dozen student formal talks, and we proudly welcome 9 of our former students, graduates, some of whom flew clear across the country to be with us today. I might add that 2 of our alumni that are here today haven't gone much far at all. They're working for Harker, and I guess that says quite a bit. We are so grateful to have 4 illustrious keynote speakers, 9 corporate exhibitors. These people bring a wealth of research and industry experience to our symposium. At this time, I wish to acknowledge the efforts of our Parent Outreach Committee, who work with me throughout the year to garner interest from our corporate exhibitors. And of course, an event like this would not be possible without all the Harker departments that I lean on to host this event. On a day like today, our facilities, kitchens, security, Office of Communications, IT departments, just to name a few, give us their personal best and remind us of what excellence really looks like. I'm so grateful for all their support. And do not forget to thank those young women in the white lab coats who are running around, scurrying, trying to keep everything perfectly flowing for you today. They've worked so hard, and I'm so proud of them. Now, just a few program notes. In order to keep things socially distanced, we've really tried to spread things out as much as we could along 3 main buildings. So you may have noticed that we have some poster sessions in the RPAC lobby. This is called the RPAC, the Rothschild Performing Arts Center. But please take note, there are poster sessions immediately next door, walking towards your left when you exit. There are dozens of poster sessions there. There's also a VEX robotics session. All of our keynotes will occur in this theater, except for one. There is one at 10:05. Yan Bing Li will be speaking in the Nichols Auditorium. So that's the other building that we are using today. If you turn to your right, you will go to our Nichols Science and Technology Center. And in the main floor, you're going to find all of our corporate displays. You're also in the rotunda going to find what we call STEM buddies. That's for our young people to play and engage in all sorts of science activities. It's also for the young at heart, so make sure you check that out. And very importantly, this year, we'd like you to go upstairs, Nichols, where we have two really fun and engaging exhibits. The first one is my anatomy students are going to be in the room just to the left of the elevator upstairs. You can go and look at the virtual cadaver, the anatomy table, and the students will tell you all that they know about the human body using this tool. If you walk further down, you'll be able to play ping pong against the robot. So it seems as though there's an awful lot to do today. So without further ado, let's get started. I'll introduce my co-president for WISCAM, Kavitha Murthy.
Hi everybody, like Ms. Chetty said, my name is Kavitha Murthy and I have the joy of introducing Dr. Kamini Varma for his keynote speaker of today. Dr. Kamini Varma is Vice President of Genetic Testing Solutions and Research Development at Thermofisher Scientific, specializing in the development of molecular diagnostic solutions for infectious disease and oncology. She began her career in genomics and has over 20 years of experience in building and transforming large multidisciplinary global teams with a focus on delivering innovative technologies and new products using qPCR, next-generation sequencing, and Sanger sequencing. Dr. Varma successfully commercialized over 100 complex instruments, consumables, and molecular diagnostic tests by taking them from concept to mass production. Most recently, Dr. Varma has been the driving force behind Thermo Fisher's COVID-19 response. She has led the development of over 30 COVID-19 products registered through the EUA and or CEIVD, currently selling in 140 countries across the globe. Dr. Varma received her PhD in biochemistry from the University of Mississippi Medical Center, followed by a postdoctoral fellowship at the New England Biolabs. She is the author of many peer-reviewed publications in parents and is a big advocate for women in STEM. So we can all put our hands together for Dr. Kamini Varma.
Thank you, Kavitha, for that lovely introduction, and thank you, Harker, for having me here to give a talk today. It is so great to actually see people in person participating, and having lived a life steeped in STEM, I couldn't be more excited to participate in your Science and Technology Symposium. I'm looking forward to all of the lovely talks and posters that all the students have prepared. So when you think about my talk today, we're going to do a little bit of time travel. I'm going to show you some images, and we're going to absorb it for a few seconds and see how we feel when we look at them. Oh. There we are. So this is on or before March 15, 2020. This is March 16th onwards, when the stay-at-home order went into effect. So what changed? We were really in the midst of a COVID pandemic, right? So before this, it was great. We went to stores, we commuted to work, we gathered at different locations, went to concerts, went to parks. And right after, we entered the state of mild panic. The stores were empty because people weren't sure how long this would continue. Streets were empty, except for a few cars that probably had essential workers going into work. Planes were empty, except for people that probably were traveling back home to their loved ones. This isn't normal, right? So what else happened? We had doctors, we had essential workers don protective equipment, wear masks. We started hearing about people dying. So you have the picture of the first woman in San Francisco that unfortunately succumbed to COVID. But the human spirit stays strong, right? We started seeing graffiti. We will survive, we will make it through, right? We will get by. Before vaccines came on, the only protection we had as a community against this virus was testing. That was the only way you could keep people safe. You could find who had COVID, get them the treatment that they needed, and then you could isolate them from people, their loved ones, sitting around them. So really, testing for the majority of 2020 was the saving grace against COVID. Now, everybody, and that then got us to our next new normal, right? So we started interacting. We started doing everything with Zoom. You had Zoom weddings, you had Zoom school, you had Zoom students, well, kind of. And then you had Zoom gatherings that were socially distanced. But this, again, isn't quite normal, right? Now, for the majority of you in this audience, COVID really started in March of 2020. And each and every one of you, whether you're a student, a parent, a daughter, a son, a teacher, have lived your own experience through COVID, and I'm sure have remarkable stories to share. What I will be talking to you today is my story. So I lead the molecular diagnostics group for infectious disease at Thermofisher. And our story began a lot before March. Our story began in January. And this is a story of a team of scientists and what they did to make a difference in the world during this pandemic. I put together the COVID-19 diaries actually in April of 2020 when I was asked to present a talk. And already by then, we had lived so many different things that it felt like, a diary or a book format really did justice to the number of chapters that we've gone through. I had no clue that 2 years later, we would still be adding new chapters. At that point, we had no clue that this would continue for as long as it did. But this is the format that, has persisted for me, and I'm going to take you through it today. So with every good book, you start with dedication or gratitude. And for me, it's been an incredible journey of thanking my team. We really, really understood the power of one team. And when I say my team, I mean the broad team at Thermofisher, because we rallied around a common cause and made a tremendous difference in the world. Around your work colleagues, given the intensity at work, it took a village for all of us to be supported in other aspects. So to my family, my husband and my wonderful kids, they were extremely gracious and extremely wonderful at supporting me through all of the trials and tribulations that I was going through, and made everything that much more special, right? So here's the setting. We're sitting around, it was week one, getting into 2020, we had returned from the holidays. I was really just thinking, okay, what lies ahead, planning ahead for the team, planning ahead for the work that we were going to go accomplish in 2020. We had started scaling up our clinical group. And then I got this email over the weekend. And the email was really very, very small and brief. It was basically that there was this novel virus that was causing pneumonia-like symptoms in China. It looked like it could be a type of coronavirus. And we had experience with coronaviruses before. There are 4 coronaviruses that don't cause anything but a common cold. There are 2 coronaviruses like SARS and MERS that had a bigger impact in the last, between 2000 and 2010. But this seemed like a new one. And what was interesting is they actually released the sequences of this, and it started the discussion of, hey, I think we probably need a diagnostic test for this. So it leads to chapter 1, which is the bat, the pangolin, and the human. So throughout the course of this talk, you're going to see different scrunched timelines. And it shows you what was happening around us at that point, right? So if you looked at the timeline on top, it was on December 31st that China alerted WHO on an unusual pneumonia. We thought it was by January 7th, a new virus, but we didn't even know if it was transmitting and how it was transmitting. It seemed like it was very contained to China, but then around I think it was February 2nd, we started seeing the first death outside of China. And really, it was on February 11th that WHO announced that the new name would be COVID-19, which stands for coronavirus disease. And it was January 30th, a whole month later, that this was declared a global emergency. So while all this was going on, we weren't waiting till February 20th or January 30th to start mobilizing around this. We were on it. And we were on it because we started hearing from our customers who needed to develop diagnostic tests. They needed qPCR instruments. I'm sure everybody in the audience now is familiar with what PCR is, or a PCR test. To run the test, you need reagents, you need instruments, you need protective, you need all kinds of materials. And at Thermofisher, we provide all of that. So we had to scale up in a massive way because the demand capitalized around that. The other thing was for my team, which had to go develop the diagnostic test, around January 10th was when these sequences started getting published. And we had maybe 40, 50 sequences. So that means somebody had taken a sample from an infected person, isolated the genomic material, and sequenced it. And you need that sequence to be able to go and develop a diagnostic test. So that's when they release these. And it's very important to get that sequence right, because once you develop the test to it, you don't want that sequence to change because your test won't work. To give you perspective, we have hundreds and thousands of sequences now in the database, but at that time we had about 40. And my team really got on it, and we developed a SARS-CoV-2 assay that was a prototype. And normally, you would have samples to test your assay on. You would know exactly if your assay is working or not. We didn't have access to samples. What we had was a test that worked on artificial systems that we knew would probably be robust. And we were trying to ship it to China at the time to just even get it in the hands of customers, that few of them who had access to samples at the time. I'll talk to you a little bit about the test. So the test that we developed is called the TACTAT COVID assay. And if you took the COVID genome and you stretched it out, what you're looking at is the gene sequence on top. You have different genes, so ORF1, ORF1b, S gene, M gene, N gene. You usually go and do PCR assays to target certain regions of the genes. So if the virus is present, that region will amplify, and you get a positive signal. So different groups were choosing different regions, and you want to find areas that are different from other coronaviruses, unique to this particular coronavirus, and also isn't prone to mutation. So those are some of the criteria you use while designing a good assay. So we in our lab decided to go after 3 particular regions, so ORF1ab, N protein, and S protein. And you'll see a variety of different vendors going after, like the United States CDC just developed assays to the N gene. Some other groups went across and, found different targets. We're the only provider that really went after S gene. And we went after S gene because it's a very conserved region, and that's your spike protein gene, and we wanted to ensure that we had that as a differentiator. This becomes very important later on, so I'm drawing your attention to that, just because at some point that's going to be a very interesting tale. You end up being considered positive if you have all genes light up, or at least two. You're negative if nothing lights up, and sometimes you can get it inconclusive if only 1 out of 3 lights up. It could be any one, and then you re-run the sample. So we had a test that had a little level of testing, nothing substantial at that point. We had shipped it to China, and most of the work at that point was really happening around China, because we thought the pandemic or the disease was really contained to that region. But things didn't stay there, as we know, as the story unfolds. We started hearing about a big outbreak in Italy. We started knowing about cases sporadically elsewhere in the world, right? And this created a whole different element of both awareness as well as call to action. Never in my life, growing up in science, did I think I would have a slide that had Vice President Pence, President Trump, and our activities on the same slide, but that's how the story of the pandemic unfolds. So this period is a period between February 14th and March 13th, where we made the call as a company to go and pursue an emergency use authorization approval. So this is an approval that the FDA, you have to go to the FDA and you have to show them data, give them studies, and get approval to be able to sell your diagnostic within the US. In a normal cycle of product development, if you develop a diagnostic assay, it could take anywhere between 3 to 5 years to develop an assay, do all the studies, run all the clinical samples, convince the FDA with the value of your test, make sure it's not causing any harm, do all of that. So that's a 3 to 5 year cycle. In our previous experience with other pandemics like Zika, it took us about 7 to 9 months, even in a pandemic like Zika, to go get that approval for emergency use authorization. But here's a tale of what happened, making the impossible possible during COVID-19. So we had decided to do a submission, and that entailed coming up with something called a pre-submission. So you come up with your whole experimental plans, what studies you're going to do, write it up, send it to the FDA. We got it done by Feb 28th. And based on previous experience, we were like, oh, it's going to take them a couple of months likely to get back to us. Well, I got a call on Saturday morning from my regulatory leader saying that the FDA wants to talk to us on Monday. Okay. So then we're scrambling inside in the teams, and we're like, okay, so what's the fastest we could do this? How could we go and talk to them? And meanwhile, on March 3rd, the other event that happened was Vice President Pence was meeting with big diagnostic labs like LabCorp and Quest, as well as big providers like Thermofisher that could manufacture and provide these tests. And the goal was really, we need solutions now, and we need to get them to the people right away. So the feedback when they came back was, we need this done in a week. And you should have seen the shocked expression of like both me and the team. We're like, what do you mean a week? And they're like, we need it in a week. And, talk about impossible. So my first reaction was, I don't even have the right level of instruments that I need for this work. And, I've taken out some of the names, but the president of the group really just went and said to my VP of operations, go charter a plane, bring the instruments here, get it installed ASAP. That's just one snippet of how we were approaching it. Nothing was out of bounds. I basically set up a war room, asked for people that beyond my group that I needed to participate in this effort. We had people flown from Austin, people flown from other parts of the world, and we just needed them there. We formed this war room, and essentially for the next 10 days, very few of us went home. We rented hotel rooms. People went, slept for like a few hours, came back. We were in there strategizing, ordering things. You have to realize there was no sample in the US that you could access. You couldn't test this. We were working with the FDA extremely closely. You'll see a lot of points where you're seeing meetings with the FDA. They were true partners in this with us, right? Talking to us about, okay, you need to do this study, this is critical, this is not. And we had access to cultured RNA that we used for all of our studies. So we were able to get that through the network, and we were able to prove out that our experiments worked. It was extremely, touch and go for a number of days on like whether we get it done in the required time frame. And actually on March 13th, that afternoon, there was a press conference where President Trump actually, Rosa had gotten their diagnostic test released, and he also said that Thermofisher was going to get it that day. And we were still submitting the paperwork, we were still getting through all of the back and forth and the edits. And we had a deadline by like 4 p.m. because it was 7 p.m. Eastern time, and the FDA was actually working late. And there was a quibble over a, slight calculation, and, the stress levels high. And finally, at like 4:05, we got all of our edits in, submitted it, and we're waiting breathlessly to know whether we got the EUA approval that day or not. This could have been a point where we submitted everything and the teams could have gone home. They haven't slept, seen their families for 10 days. Nobody wanted to leave. We wanted to know if we got our EUA approval, and everyone was staying put. Well, around 6:30 that evening, as we're all sitting around in a conference room, we got the letter from the FDA saying that we got our emergency use authorization. And you can totally see the energy and the jubilation in that room. And what is exciting to me is this is a team that is still all present at Thermofisher, and they're still working on this to this day. So it was really, really nice. So we finally go home. We have a test that is approved. So for all of you, what was the test? So it was basically a sample that could be collected, a nasopharyngeal or a bronchial lavage. We would then do sample prep and isolate the genetic material RNA using Kingfisher Flex. You would then have an assay that would do PCR and give you the output on an instrument, which is a 7500DX. And you had an interpretive software that would tell the physician whether it's a positive or a negative. So that's your solution that got through an EUA approval. So of course we did this. We're happy, we're going home. We have plans to go and sleep for like 10 days and never talk to anybody. Does it work out that way? No. Right the next morning, I got a call from our manufacturing group, and they're like, how are we going to release the product? What is the specs? How are we going to take this and now go and register it across the world, because this was just the US, and so on and so forth. So we're back at it, right? And this is, the next chapter is really a story of scaling. And this, again, is March 14th to April 20th. It's a month. It's living 80 lives in a month, right? That you would normally, again, take years to do this work. We had the assay that had gone through EUA approval. You could sell that in the US. What about the world? Because we're a global company. Our mission is to provide solutions worldwide. One of the other key registrations is a CIBD registration that covers a lot of Europe and Africa and other places, but we and the regulatory team really went worldwide. We kept registering the product across 140 countries, and that allows us to provide our solutions to anyone and everyone in the world. We also had to continue evolving. So I showed you the first solution, which was on our 7500 instrument. But some of our customers had a different model of instrument. So we had to do a lot of the work to enable that. We were dealing with supply chain constraints. So some of our vendors couldn't provide certain materials. So we had to test out new materials to be able to swap them out. And each of this requires studies and resubmissions and work with different regulatory agencies. And the team kept building. They kept adding more and more capability. We also had to provide material. You are now scaling to provide solutions globally. We had to buy raw materials. This is a polyethylene glycol. We probably could have done like a month's supply or a year's supply with one of those cartons before. We started buying things up in bulk and housing them in warehouses. We built new warehouses. We built new factories that could produce VTM because VTM was at a shortage. We just went all out on the global scaling. We hired people. We became, in a lot of cases, I had R&D leaders going and working in operations to sometimes label products or sometimes build products. We were everywhere, right? We started working with governments and agencies, and we started working with different institutes to help set up. I'm showing you an example of the work we did with the UK government. The UK has a nationalized healthcare system, and they really went all out and built a lighthouse lab network, set up in 3 different locations between Milton Keynes, Manchester, and Glasgow. And the idea was that would become their centralized testing facility. And, they really started with empty labs. We were running short of, we couldn't provide instruments fast enough across the world. So what they did was they went across to all of their labs, whether it was academic labs, research labs, all around the UK, and gathered up all the right instruments, the 7500 that had been cleared. They had their army called in to mobilize around this effort because we were inherently fighting a war. And that was all of our mindset. We were in it, we were fighting a war against the virus. We sent out field people. So you have to imagine, while everyone is home, quarantining, isolating, there is a whole slew of field service people and scientists who are out there going to customers, still trying to help them and make them successful. We got those set up within a weekend, and we got them running. And the UK actually went on to do hundreds of thousands of tests per week and had one of the best testing programs in the world. So to give you a perspective of the mindset, right? So we've launched this, we're all feeling excited. In a normal cycle, like I said, you would test the solution for years and years before you released it out. Here we released it out to like the whole world, and we still hadn't got our arms around enough data from real samples, right? So there was a little bit of nervousness in the team, like how does this perform in the real world? And this was in April, and we started seeing feedback. UC Berkeley had set up a whole testing lab, and this was a quote from Jennifer Doudna, who's their a premier researcher there, basically testing different solutions and really calling out ours as the most sensitive, and that's what they implemented. We started working with different governments, like I said, even within the US, we had a whole program to empower testing and enable testing in different states. And you know, a funny anecdote, right? Like in a different world, you would get a call from a customer, or if they weren't happy or they were happy. Here, you never knew who was going to go and give a press conference that day. So you could have a governor speaking, or you could have a customer speaking, and everyone, of course, is a COVID expert by now, right? So it was an interesting time because, you know, it's a very different way of thinking and doing science in than how scientists are generally trained, right? So we surely must be done by now. We had a solution registered in 140 countries. We were providing the material. We had it broadened out to a number of solutions. So, you know, what's left, right? Well, a lot. When customers were scaling up, what we found is one of the pain points is all of these labs were really set up to run maybe a few hundred samples a day at best, right? They're now dealing with thousands of samples a day. They were set up to have trained professionals that had been trained for years. Now they were picking people off the street and having them run. Trained professionals were falling sick, or family members were falling sick, so people were quarantined. So you kind of had this crisis around, like, we need to be able to run thousands and thousands of samples a day, things that we've never done before. And so, you know, another conversation with the president of my group, and you know, he just casually throws it out there. Hey, what do you think about building a solution that could run 10,000 samples a day? The Kamini from 3 months back would have probably said, it would take me like 4 years, and it would take me like, you know, it's not possible, or any of that. The Kamini of April, by then, was so used to these curveballs that I was like, sure, let me figure it out. And we'll come back with a plan. And that's how our mindset, even already by then, had completely shifted to how do we tackle these things that we have never actually dealt with before. So we developed a solution that is in the market called Amplitude. So this is a slightly longer timeline, May to September, because it's actually a very complex system. It's an automated system that requires a lot of interplay between from start to finish. But you know, the team went through, and again, we had to clear out labs and build new facilities in order to house these systems and drive to that. And again, we were, by the way, we ourselves were hiring talent all along because we needed more and more people with different experiences, and we were hiring and we were building, and people were joining the team while developing this. So it took us 138 days from concept to launch for Amplitude, and it got an EUA approval, it's got CIP approvals. We had, just to give you a magnitude, to ship one Amplitude takes pretty much half a plane. And this was the first time we had done something where we would assemble the instrument at the customer site. Kaiser was one of our first customers, and they had ordered 13 Amplitudes. And the amount of work it took to get the site ready for them and get the instruments shipped, the FAS, FSC, R&D personnel that went on site to get them running was pretty spectacular. But they were up and running, and so were a number of other customers very soon. How did this have an impact? I just show one example. So the examples that you hear about are through all of our, sick people testing, but there's a whole back to life element. The Olympics was the single most big event that has happened in the last 2 years. It's really important to have these events. I mean, all of you students, kids, you spend a lifetime preparing for this. If you miss an Olympic for an athlete, it could mean the difference between actually being a gold star or never participating, because they might just be out of their prime. It's 4 years of intensity for them to get there. We were able to support the Tokyo Olympics because what was critical was they needed the ability to gather the samples from the athletes, from the workers that were working there, and get an overnight result. And keep continuing with this. Our solution met their criteria, and we were able to get some of these big events going, which is a very proud thing for us. While we were developing Amplitude, we were also getting ready for a winter season. So you have the flu coming up. We're all familiar with the flu. But, the flu, COVID, all of these have very similar symptoms. So the prediction at that point was, hey, we need to really be ready for the flu season. So you need another test that can differentiate between flu, influenza A, influenza B, and in Europe, we actually included RSV too. So we developed this test, went through all of the registrations, all of the countries, all of the platforms, and we kept building. Thankfully, we didn't have a great flu season that year, but the solutions needed to be ready because we were always 2 steps ahead, or we needed to be. We continued diversifying. There are a lot of people that could not make it to labs, couldn't travel to get a test done. We wanted to take tests to homes. So we partnered with Everlywell to do home testing, and we managed to get EUA supplements for that. So we kept expanding. We kept expanding to create the capability to come back to schools, to come back to work. We partnered with Color Health. Each of these partnerships requires you to validate the workflow with a collection, partner with them, take it through all the registrations, prove that you can do this, and then enable it in the population. So these are just a few examples of how we've continued expanding and moving forward. I showed you an example of the Tokyo Olympics. That was a worldwide event, right? This, for me, is personal. And this was a letter I got from my sister, whose mother was my aunt, who's in India. I grew up in India. And when you grow up, you love science, you follow a path, you never know where it's going to lead. During COVID, I was working, we were all head down, we knew we had read, we knew that. But I got this letter and I was like, hey, they did a COVID test and her test was negative. And then what was interesting is it's performed on the TACPath combo kit. It was such a great moment because, your work has actually reached someone that you love and care for, and you didn't really put those together before that. So, these are the moments that really make you that much more engaged and passionate about what you do. So you've heard me talk about the COVID diagnostic test. You heard me talk about our TACPath assay, which we've, all of our solutions products, we've supported about 50% of the worldwide testing that has happened on COVID. So that's a huge impact. But what we weren't expecting and what happened is a tale of two tests. So we developed the test on COVID. We're working closely with different governments, different groups. And we work closely with the UK, right? And we're talking to them and they're like, something's funny is going on. We're starting to see the dropout of your S gene. It was 0.1% at the time, 0.2%. This was back in September, October of 2020. And we're like, that is odd. Everything else looks great, that is odd. And we all got into it and we started looking at it. Come December, that number was just shot up. It's going up to 10%, 20%. And weekly, it's just out of control. And it turned out it was the alpha variant, because in the alpha variant, in the S gene, when I talked about vaccine manufacturers going after the S gene because S gene is conserved and we're the only test with the S gene, when you start introducing vaccines, the virus tries its best to evade those vaccines. The virus does what it does to make sure it's changing form. It wants to survive, right? So this particular S gene region became very prone to having mutations. And that actually knocked out our assay. There was one particular deletion in there, 6970, that went and knocked out our assay. And because it was so dramatic, we could then take those samples, go sequence it, and say, oh, it's a whole new variant. It's no longer your original COVID. And what is interesting is you can see the path of travel for all of these variants is different in different countries, where we seem to track a couple of months after whatever happens in the UK. It's been our general experience. Brazil had a whole different variant at that time, P1 originally, and South Africa had a different variant. But what has been interesting and ironic is over time, we all kind of migrated to Delta. Delta became a primary variant worldwide. And now it's shifted and it's Omicron that really has taken over the world and become omnipresent, right? So I talked to you a little bit about the alpha variant and how we detected it. It's a little bit ironic and completely not something we could have predicted or planned for, but it has played out in such an incredible way. When alpha started becoming the predominant variant and when delta came about, we no longer had the S gene dropout. So you now went from a 2-gene assay back to a 3-gene assay. So this started coming back and researchers are reaching out, oh, there's something funny going on. This is coming back. And they go and sequence it. And they see it's come back. So now we're back to our 3-gene assay. Fabulous. Omicron hits. Oh no, South Africa. We lose the S gene again. And Thanksgiving 2021, on the day of Thanksgiving, I get a call. Hey, Dr. Tulio Oliveira from South Africa is seeing this and he wants to have a conversation with Thermofisher tomorrow morning. Are you available? So we talked to him and he's like, oh, this started happening and we saw this. And this was Omicron. This was even before it was announced that it's a variant of concern. When they sequenced it, they saw 28 different mutations. It's significant, right? So, that again became a proxy worldwide because sequencing takes time. This is, labs that are running this can start seeing and picking up new variants much more quickly with this. With BA2, we're seeing the opposite where we don't have this. So now we're going back to this. And, one of the things that we have started monitoring, again in South Africa, is a new BA4 or 5 variant that is starting to emerge there. And time will tell if that actually takes over. But that's kind of been, we developed a test that not only serves as a tremendous diagnostic, but also has become a proxy for surveillance and understanding the migration of the virus as it travels through different countries and different regions, which has been extremely beneficial in countries getting ahead quickly of knowing what is going on. We also have partnerships in the US between the CDC, the NIH, private labs. It's a RADx partnership for Project Rosa. There's this tracker that is accessible to everyone in the world and basically tells you what mutation is happening in which state and how it's traveling. So it's a little bit boring right now because mostly everything is BA2. But as you start seeing new ones come on, you'll see how it shifts. So this is part of our efforts in supporting surveillance in the US. None of this that I talked about happens without people. People are essentially the core of it, right? We have an incredible team. And, you know, when you think about words like, this is impossible, that got taken out of the dictionary. When you think about things like, oh, that's going to take forever, it pretty much, our mantra, and this is a line we've repeated 1,000 times probably by now, is a week is a day is a week is a month, and a month is a quarter. And we would literally plan like that. Like you couldn't think 3 months ahead or 6 months ahead back in 2020 because that was like, you know, thinking 5 years ahead and predicting what would happen. It literally was like you had to move with it right then. It sounds like a great story. There were moments of sheer stress and panic and worry. But you have a brilliant team and you have a dedicated and resilient team. So we hear things like growth mindset, we hear things like resilience. That is what you need to survive this. We hear things like adaptability. Nothing you predict will ever be exactly the same ever. So how do you, like, you know, when faced with a problem and you can't think of a solution, you're not alone, you have a team, use the team. When you think about like, hey, how do I solve this? Change the seat you're sitting on. Look at it from a different lens. Come up with solutions that, you know, just seem crazy, but they're actually solutions that work. So that is what I think we learned going through the COVID journey. From a Thermofisher perspective, our whole mission in the company has been around providing solutions. We're 100,000 people, company with a lot of different solutions, but we pivoted in a big way to help our customers, help patients, help people. Whether it was providing lab equipment, whether it was providing, you know, our instruments, our testing solutions. This is the only part that I've touched on. And we've really supported the clinical trials and the development and manufacturing of vaccines. So whether you're taking the Moderna or the Pfizer vaccine, we help with the manufacturing of those. Our mission really has always been to enable our customers to make the world healthier, cleaner, and safer. And with COVID, we have lived that every single minute of every day. And really, if I think about my team, this is my R&D team, right? It is not one biologist, it is not one scientist. It is a whole bunch of different skills, whether you're a bioinformatician, whether you're a virologist, whether you're a software engineer, whether you're a mechanical or an optical engineer. You could be an algorithm developer, you could be a biostatistician. You could be a molecular biologist or a chemist or a bioengineer. This is the group of people that made this solution and this range of solutions possible. This is the R&D group. there's a whole layer of marketing and business people. There are finance people. There's supply chain and operations people. There are manufacturing people. There are field application specialists. There are tech support people. It takes an army, it takes a village, and the whole village bandied together. When I look at this room again and seeing students, you all are going to be the next generation of a virologist, a software engineer, an AI ML expert, right? And we know we're going to have more pandemics. We know we're going to have more things that hit us that we have never ever thought of. And you will be the generation that will be solving for those things. And the future is in your hands. And I cannot wait to see how it unfolds for all of you. So thank you for listening and giving me the time today to come and present my story and our story, and happy to take any questions.
So much, Dr. Varma, for that inspiring talk. So from the slide on the side, we'll be answering a couple of questions. Unfortunately, we only have until 10 o'clock, so be sure. On the question, there's a little like button on the side. We'll be answering the questions with the most number of likes, so if you want any question answered, be sure to hit that button. So our first question is, I think it's referring to the first assay you discussed. How did you know that assay would work?
Because I have a brilliant team. That would be number one. So, you know, we have design principles governing how to build a good assay. So, like I said, a couple of things on the assay design of how you pick the right regions, how you know what parts to avoid, how to design the sequence. We have a lot of like proxies, so synthetic templates and other things that we use in order to characterize the assay in the beginning. We do a lot of in silico analysis to make sure there is no cross-reactivity or, you know, so the assay is very specific. You can also buy, right? Like, so what we bought was genomic material for all the other coronaviruses, influenza viruses, and you test against them. So you want to make sure you don't pick up any of the other material that is out there. So that's your cross-reactivity and specificity. You have then sensitivity studies that you do, and you take these artificial templates and you test them, and you see how low you can go while still detecting it. You do things like, you know, spike in genomic RNA in matrix, so whether it's nasopharyngeal and things. So you look for inhibitor tolerance, you look for things that like, you know, either block your assay or still make it work and give you the sensitivity. So there's a whole bunch of, between in silico design, using artificial templates and genomic material from other viruses that let you get to a point where you're very confident. And just, you know, we're a company that has been doing this for 20, 30 years, right? So we have a huge track record in doing this, and we have millions of assays out there already for many, many, many different types of targets. So the only gap was we didn't have real samples in the beginning. And this actually has stood the test of time beautifully.
Okay, our next question is, what is the main difference between 2 or 3 variants, say alpha and delta, for example?
So each variant is different from the other in what types of mutations it's carrying, right? So there's not like 1 or 2, like, you know, the constellation of variants is different from one to the other. Variants happen all the time, right? The only difference is some variants actually gather momentum and are more transmissible and take over the population, they're stronger. And then there's some variants that pop up but never gather that level of speed or that level of capability, so they die down. But variants happen all the time. It's just a mix of mutations that are different in each one that classifies it.
Thank you for the answer. Okay, next question is, what was the biggest challenge in creating the assay?
Time, right? You're in a fight against time. So, technically, is this doable? Absolutely. Is it doable in a minute? That's where the challenge lies, right? And we always knew we were in a race against time. We were in a war. We were in a daily war. It was never, and we still are. We're still 2 steps ahead. We're still 2 steps ahead of surveillance and things like that, right?
I think this is a little more of a personal question. Can you tell us about your own research journey and career journey?
So I'll need an hour for that. Kidding. I grew up in India and, really just always loved science. And I had a bachelor's in genetics, came here for graduate school, did my PhD in biochemistry, and I always knew I liked applied science. So I never wanted to go into an academic career. I chose to do my postdoc at New England Biolabs, which is industry postdoc. And I always liked applications or putting products out there that could really impact or change things that are very visible, right? Like in academia, it's like, yes, you're doing a lot of very important research, but it is focused on solving one problem. Here you have a breadth of offerings. So I've just always just followed my passion, taken opportunities wherever, never shied away from doing difficult things, and you find yourself doing amazing things. And
okay, how many people total did it take to produce? I'm assuming it's talking about the assays you developed and maybe a little bit more about your team.
So hundreds of R&D people, right? So we started with a smaller nucleus, probably close to like 50, 100 people in the beginning. But then by the fact that we were proliferating and growing our list of products and solutions and things, it takes hundreds of different people from different skills. And you have to keep in mind, this is not just the experience that is built in over the 2 years, right? These people have been working in these fields for 20, 30 years, many of them. So you're coming with that collective wisdom and that collective knowledge. And when you ask me, what made us sure that we were going to be successful, it's a lot of that. It's your intuition, it's your collective knowledge and your gut, right? And then if you think about the broader teams, it's thousands of people. Yeah.
Okay, another a little more about your background. Being a woman in STEM, what has been your experience navigating a male-dominated field? Have you ever worked to overcome the barriers as a female in your path?
Definitely, right? It is, as much as the momentum is changing to more and more women in STEM, it's still, not as, or at least 20, 30 years back, there were very few women in leadership roles. There are very few women still. There's a lot more scientists now. I think it's, again, just be true to yourself and follow where your heart goes. Be strong because you have a voice. Use it. Your voice matters. Your voice is no smaller or bigger than anyone else's who wants to be in this role in this game. And be brave.
It's a great answer, especially for women in STEM or in the audience, all of our STEM members. Okay, next question. How have you seen the health policy shift in different countries as a result of the vaccine and the virus?
Yes, this is an ongoing evolution, right? So I think with COVID, there's a realization that things happened globally. So it's no longer what happened here is staying here, right? And people travel very easily, viruses travel very easily, so the proliferation is just very different. I could see a lot of discussions around global surveillance and global data sharing, standardization of data, because how do you compare a test? Not all tests are created equal, so how do you compare results from one test to another? we're in a lot of like discussions with, different groups around with like the FDA, CDC, different governments. I think there's going to be a momentum towards data sharing, data standardization, and how we all use real-world evidence to bolster our studies. So there's a lot of that. New technologies coming up and how they play a role in where we move forward.
Thank you for that answer. Okay, next question is, are we done with COVID-19? Assuming this is referring to your work on the virus, and if there's any more new projects that are being developed.
So I'll actually steal a quote from my medical director on this. Are we done with COVID-19? We think not, right? So we think, and this is his quote directly, it's like we're top of the fifth inning when it comes to COVID, right? So for all of you baseball fans out there, you probably can relate to this more easily. But when COVID originally hit, people were really impacted. We didn't have medicines, we didn't have an understanding of the disease. Then Alpha hit, and it really impacted the UK in a significant way. Delta came, it impacted India in a big way because they didn't have the vaccines and things. Omicron was the fourth wave, and it really has had a much smaller impact because we're that much more prepared. We have vaccines now, much more global. Yes, people have gotten affected, but the number of hospitalizations, the number of deaths have been lower. So I think we've caught up, and hopefully we'll stay there. But we're starting to see BA4, BA5. There's still lineages of Omicron, but do we think it's completely done? Probably not, but time will tell. One thing I've learned with COVID is never predict it because you're going to be wrong right the next day.
I think this is going to be our last question for this talk. How do you think the COVID response at Thermo Fisher will help in the preparation for any other pandemics to come?
Oh, we have learned a lot, right? And we have learned that we can go solve anything when we put our minds to it. So at Thermo Fisher, the capabilities that we built and just the mindset around this, we believe, carry over to all of our clinical products as we move forward and all of our solutions. We had, for a big company, we had a very unified and singular mission, and that's always, I think, key to having a successful outcome.
I think that's all the time we have for today. Can we get a round of applause for Dr. Varma?